on with our next speaker, Professor, Le Song, who's, and just give me a second here. Yes, Professor Le Song, who's also the chair of the machine learning department here at MBZUAI. He regrettably couldn't be here in person, but he will be giving his talk virtually over the Zoom system here. So, Le, are you connected and ready to present?
Yeah, you can hear me well.
Can we up the volume a bit? We can hear you. We're just going to do a little bit of volume adjustment. Can you try saying something again?
Can you hear me?
Perfect, right. And Professor Le Song's talk is going to be on AI systems for drug design. So it's all up to you now, Le.
Thank you for the introduction. It's a really nice workshop. I'd like to actually ask a question instead of, explaining AI system. I'm asking question, what will be a very good scalable system for drug design? What I'm going to do in this talk is sharing you my experience in the past year building AI model, a family of AI model for drug design, right? Just to give you a little bit background on drug design. So it's really a time-consuming and costly kind of process. So typically, to develop a drug for a particular disease, it might take 10 years to do so. So one of the reason is that the space of drugs are very large. So for instance, if you're looking at a small molecule drugs, it might involve 60 actants, and then the space might be 10 to the 60. So you need to find a few compounds in this huge space, which is going to be effective drug. So actually, there's a very rigorous process of selecting from these huge space. So roughly in the drug design industry, there are 3 stages. For the stage 1 is going to be the in some sense selection process inside the laboratory. So you can use a different type of high throughput technology, machine learning, data analysis to narrow down the number of candidates to something like a few hundred. And then you go into stage 2 to do further selection, narrow down to a few using, animal experiments, right? And then you move to stage 3, which you go for a real patient clinical trial, right? There you might have a sub-stage as well, right? Until in the end, everything is good, meaning that the drug is effective and is not toxic and it has significant improvement over previous drug, things like this. And then it gets approved by regulatory body and you get one drug and then pushed to the market, right? So besides this huge space and then 3 stages of selection process, in every stage there actually could be some failure because every stage needs to be validated by experiment, wet lab experiments. There may be failure and candidate suggested by the earlier stage might fail, all of them may fail in the experimental stage. You need to go back and do the selection again. So the process is really long and time consuming, right? And besides long and time consuming, and the cost of developing drug is very high. Imagining, you have a 10-year process of developing one drug. Just think about the number of experiments involved and the number of scientists involved, the number of assay computing power involved. It's huge. Especially you go to more complex kind of diseases, the cost of developing one drug is becoming increasingly high. For instance, during the 1950s, if you spend $10 billion, then you might be able to discover a few drugs, right? Okay, right. But as time goes by, right, some of these easy to develop drug or disease has been already cured and treated, right? Now, what's left are those complex diseases involving tumor, cancer, and it's really hard to, figure out what is the right target and what is the effective and drug for them. So after 1990, so the cost of developing one drug is around $1 billion, right? So, so the question is, if we can use some of these data analysis approach, AI-based model to reduce the times of developing the drug and reduce the cost, right? If we can do that and automate the process, we might be able to accelerate the speed and of drug discovery and bring down the cost by a lot, and more disease can be cured and more people can be helped, right? So why is such a problem so difficult? It's mainly because we're really dealing a very complex system, right? So in some sense, human body is a multi-scale heterogeneous network. So you can think about it in a causes level. Our human body is going to consist of different organs. They coordinate and then collaborate to achieve the function of the human body, right? If you actually zoom into each one of these organs, for instance, the lung, then you will find that it's actually a network of different type of cells. And these cells, they are not isolated from each other. They talk to each other through cytokine, chemokine, and then cell-cell membrane protein interactions. In some sense, you will find that there's a network of cells and they're interacting with each other. So if you look at a particular cell and zoom into it, there's again a very complex network. So inside the cell, there are all different kinds of proteins and DNA, RNA, and some small molecules. They interact with each other to carry out the function of that particular cell. So within the cell, there's a very complex network. If you zoom into individual molecule inside the cell, for instance, a protein, it has its own structure sequence, and the function is closely related to the sequence and structure of that protein. And we need to understand also, the structure in order to be able to design drugs. So in some sense, in order to understand the disease, you have to look at this multi-scale system at all different levels, right? And then in terms of target discovery, is really to find some important node or important, locations which can differentiate disease, for instance, individual from normal individual. And it's also very important to discover those important points where you can regulate and steer, for instance, disease system against its normal system, right? So once you identify those points, of course, you need a way to be able to steer the system to change the disease states. That's about designing drugs for that specific target. Then you have to go into this molecular data in order to design a small molecule or a protein to be able to intervene and steer the complex multi-scale system, right? So fortunately, biotechnology has advanced so much that we get data from all these different scales. For instance, when you look at the target discovery problem and look at the organ level, intercellular level, and even cellular level, there are many different ways of measuring data. For instance, we heard a lot about genomic data, epigenomic data, transcription data, proteomics data, and metabolic data. It's all about, data related to the expression or the data about a particular cell, right? And besides the molecular level expression data, you can also find that there are lots of high-level approach to collect or measure the interaction between these molecules. So this molecule form a complex network. Part of this complex network has already been figured out by biologists or by technologists over the past year. We have been able to, accumulate this type of the multi-omics data very rapidly. Not to mention, those image data we are very familiar with, the increasing amount of image data in this healthcare record data, which you can use to analyze the phenotypic differences between disease and normal, right? So when it comes to, so essentially what you want to do is to take all this data, right? Molecular level data of different type of molecule and the interaction between these molecules, and then going even a few level up about cellular level, cellular system, tissue level kind of measurement to be able to discover what is the important points or node in this complex multi-scale network, which either differentiate disease from the normal kind of cell or tissue, or there's an important node where you can modulate it, control it, intervene it to be able to change the status of the cell or tissue, right? So when it comes, once you have identified the important node in this very complex system, it's typically going to be some kind of protein, right? And then you have the designs and jugs specific for this protein. So as I mentioned earlier, this protein in the cellular system is going to be having such a complex 3D structure. So the 3D structure is very important for the function of the protein, and designing a jug is also critical to take into account the structure of these proteins. You can think about the structure of the protein is going to be the shape of the lock, right? The lock hole, right? And then the jug is like a key. It needs to fit into the particular lock hole in order to open the lock to treat the disease. So you have to collect lots of data about a protein sequence, protein structure, and also the structure and sequences for other molecules involved in the system, such as RNA sequence, RNA structure, molecule, kind of chemical composition, and 3D structure molecule. So you will find that there are lots of data involved there. We are already haven't done in terms of sequence data, structure data, they're all there. So there are lots of machine learning problems for this type of highly structured data, right? As you look at the current databases, and over the past year, I have been also working with some engineering teams to curate and clean up this data. And the scale is already quite big. If you look at the number of protein sequences, it's already a billion level. And if you look at the number of protein structures, it's already 100,000 level, right? And then in terms of the number of interaction between these proteins, is already tens of billion, right? 100 billion level. So we have also been able to measure the gene expression level of cells. And if you look at the available ones in the public space and curate them, you can easily reach 100 million level single-cell and seq data. Also 10 to the 6 millions of perturbation data, lots of publicly available images of different types of cells and cells under perturbations, cells in different tissues, right? There are also huge literature describing, different disease, different type of tissue, and different type of gene regulatory relationships, right? The data is quite diverse and huge. And if you look at the speed of the increase of the data, it's also going to be very fast. So these plots on the right here, essentially the blue line is going to be the speed of the Moore's law, right? The speed of the doubling of the shape density, right? Moore's law, and then the growth of these data in the space of biology or medicine, and then computational biology is much faster than that, right? Okay, so in some sense, now we're at the point where data for drug design and computational biology are huge, right? If you just use typical bioinformatics tools and using manual analysis by bioinformatics expert, so it's very hard to catch up with these scales and growth of the data and the speed of the data, okay? So in some sense, you will need some kind of AI model to be able to first integrate this complex data at different scale. The model or system has to be scalable enough to be able to ingest these large amount of data, and it has to adapt as we get more data, right? Another very important thing in the drug design industry is you need experimental data to evaluate your model performance. In some sense, experimental data is going to provide the gold standard measurements. Previously, in biological kind of experiments, discovery, or in the drug design industry, the experiment part and data analysis part are hugely overlooked, right? Open loop, right? So typically, the experiments are driven by a hypothesis by some expert, right? And the expert may pre-design a few hypotheses, and then the experiment conducted according to the hypothesis. After maybe 3 months to 6 months of time, or even longer, you get the results, and the data typically comes in Excel table, and then it's analyzed by human, and then the result returned to the expert to see whether the hypothesis is validated or not, and then the next round of hypothesis is designed and experimented. So the whole kind of cycle is very long, lasting about 6 months or a year. It really has very few iterations between the model data analysis part and the biological experiment part, especially in the case when the biological space you want to experiment with very large. For instance, the number of memory proteins, 7,000. If you want to look at each one of them and look at the combination, the space is very large, and you want to carry out perturbation experiment, then there's a huge number of perturbation to do, and you cannot run this iteration for too many times in some sense. If there's a way to somehow have a system integrated better with the experimental system and drive the process in a faster, more efficient, automated way, then the entire validation process or discovery process could be potentially be further reduced. So given the above context, I've been thinking and has been working on this for the past few years, how to design a system, AI system for drug design, right? At the moment, I've come up with some design, which are more of a family of machine learning models. So I'm very interested to talk to the expert in this community and see whether such AI system can be, made more scalable, efficient, such that it can better support this, drug design process. So in fact, the system reflects the complexity of multiscale nature of the human body in some sense. So you can remember two lines of the system. One is the family of 4 nested machine learning big model, right, pre-trained model, and then there will be 2 closed loop components, right? So in this nested 4-level system, the innermost is going to be machine learning model for protein, right? Yeah, that's very important for designing drug molecules. And on level above is going to be protein interaction. The protein never function alone, and it always interact with other proteins. And when we're designing drugs, we are also designing some drugs to interact with the protein. So we had to be able to very accurately predict how protein can interact and the location of the interaction. That's why one level up is protein interaction. Another level up of is going to be model for a cell. A cell consists of a network of protein interactions and protein regulation, gene expression, and many interaction of different type of molecules. So you want to take into account that interaction network, and also being able to integrate these multi-omics data to be able to make prediction about how the cell is going to respond to perturbations to find the targets, okay? And one level above cell is going to be cellular system. The cell never exists alone, so you always interact with other cells as well to carry out the function, and cells interact to form tissue as well. So in some sense, you can expand it further. You can expand, for instance, model a system for tissues and even to the entire human kind of body level kind of model. But this 4 nested level of a system is going to reflect most of the part of drug design process and target discovery kind of process. This system is going to sit on top of these large curated data I just mentioned to you, okay? Right. I'm going to expose later a bit more about the detail of each one of these machine learning model and explain some of the dimension of this model in terms of number of parameters, some of the current effects we achieve within a smaller model and by consuming smaller amount of data. So we hope by consuming the entire set of data and with larger model, we'll achieve even better results, right? So this is the four nested modeling system, right? So we think necessary for a target discovery and drug design. Besides this, we have a two closed loop component which is needed. That's the closed loop between the machine learning system, AI system, with the experimental platform. The first level of closed loop is going to be, experiment system on cellular perturbation and cell system perturbation. And we want our target discovery kind of component involving cell and cellular system perturbation to be in closed loop with this experimental system. And you have to use some kind of active learning kind of approach to make the machine learning system and experimental system to closed loop to reduce the number of experiments, right? Another closed loop system is going to be in the drug design kind of pipeline, where you want your prediction or design about protein drugs or small molecule drug to be experimented in wet lab system as well. Again, you want such a system to be closed loop, and a machine learning system is able to generate hypotheses very quickly and send to this wet lab system to do the experiment. Active learning, again, is going to provide a very important tool in between to reduce the number of experiments and quickly optimize your entire drug design, right? So now I'm going to zoom into the detail a bit to explain this four-level system, hopefully exposing to you more parameter about the scale of this model and complexity of this model, right? And then you have some kind of material to think about in order to build a large scalable system, whether there already something out there or whether you need to design something new to accommodate this family of nested models, right? First, I'm going to zoom a little bit more into this target discovery aspects, which involve mostly the cell modeling and cell system modeling, right? So the kind of a little bit more detail about this, the cell model is going to be something like this. So the input to this cell modeling kind of system is going to be multi-omics data. Those data about gene expression, protein expression, and then also in some sense, the protein interaction network or protein or transcription factor related gene expression, right? These interaction networks is going to be, either obtained from the curated data from previous experiments, or one can run natural language processing to extract more from the scientific literature, or you can also obtain some of these predicted edges based on protein interaction predictions, right? So in some sense, this network here is going to link together those molecules, entities you can measure further using this multi-omics data. So as I mentioned, that in the space, there's already 10 to the 8, 100 million kind of single cell multi-omics measurements. Here, you don't have any label attached, but you can think about it. These are the sentences out there in the internet, right? You can actually pre-chain, in some sense, a model to represent those molecules involved. You can pre-chain a model to represent, for instance, the genes and proteins in the system, right? Here you have some kind of large-scale pre-trained language model kind of task to ingest this multi-omics data, right? So you can think about there are 10 to the 8 sentences, and for each one itself, it's going to be something like 20K genes, and it's like the sentence length is going to be 20K. It's really a long sentence. And there's also a very complex graph linking these 20K tokens together, right? So you have to develop potentially some new transformer or graph transformer model in order to be able to pre-train on this type of data, right? Once you have representations on these genes, then you want to be able to encode perturbation as well. And there's also quite a lot of public data out there for perturbation. After you have encoded perturbation, you really want to use this model to adapt to a particular type of cell you are interested in to make prediction about perturbation. and identify targets. For instance, if you're particularly interested in immune cells, then you have to, in some sense, adapt the model to immune cells and be able to predict what are the perturbation to immune cells, which can trigger the activations of the immune cell, right? So, so these are the kind of the modeling kind of the system you will need. So in order to measure the activation of immune cells, the measurement may be images or other kind of biological assays. Then you might also have the need to process the images of those cells. Then the computer vision techniques may be needed in this case, right? Beyond single cell, then it's going to be cellular system, right? So cells typically need to also communicate with other cells to carry out its normal function. And when you're experimenting with these cells, you might also experimenting co-culture, two different types of cells. The perturbation may be in the one type of cells. It can propagate to another cell and affect the function of another cell. Very often we need to build a model which can, in some stitch together the model from the single cell to be able to make a prediction about how cellular system changes. In some sense, the modular kind of nature of this cell model is very important to be able to stitch together, this different component to be able to make a prediction about the cellular system. Again, for each one type of cellular system in a lab kind of environment, you may be able to do around 10K perturbation for each one of the system. And then you want to adapt your model, pre-trained model to be able to be quite accurate on other perturbations, yeah? So based on such architecture, we have tried a smaller kind of model and consume a small set of this data I mentioned and use that to build some kind of a cellular perturbation model. So the model we build based on this pre-training system is already improving over SOTA by roughly 10%, right? This is as you increase the training proxy of the model of this pre-training system is able to show quite a big improvements over state-of-art model. So now it's achieving something like 82% accuracy in terms of the perturbation prediction, right? Yeah, comparing to existing ones is like 71%, right? Using such a model, you can imagine I can also make prediction about new type of cells, what kind of perturbation is going to be effective, right? We find that here is a concrete example, right? There's one type of cell called the fibroblasts, and is closely related to this IBD disease. And one of the well-known targets is IL-13 receptor. And using our model, we are able to find other targets which are potentially more effective than these well-known targets. And one of these targets we have been sent to further evaluation for it, okay, based on such a pre-trained model system, right? Next, I would like to zoom a little bit into this protein kind of pre-training model and protein interaction prediction model. So that's actually related to one of the recent advances in protein structure prediction, AlphaFold 2. So, but we have made a significant improvement and modification to the AlphaFold kind of architecture, especially in terms of bringing in this pre-trained model. So as I mentioned earlier, there are 10 to the 9 billion level protein kind of sequence data out there. What AlphaFold 2 original was doing is it's not using a pre-trained protein language model, but using some kind of multiple sequence alignment to be able to utilize this sequence data to, in some sense, design some specific feature using this large library. So what we do is we actually apply a protein language model, okay, mass language model on top of protein sequences to be able to learn some kind of sequence representation for proteins. And these type of embeddings from the protein language model is going to feed into some kind of cross-modal transformer and structure prediction unit to be able to predict the structure of the protein. what we find is that as we increase these pre-trained protein language model from 100 million to 1 billion to 10 billion model parameter, so the downstream structure prediction model is actually able to improve consistently. So the next step we are planning to do is to train an even bigger protein language model, something like 100 billion. Hopefully we will be able to better ingest these billions of protein sequences into this model and being able to perform very accurate protein structure prediction, but much faster. Okay. Besides protein structure, predicting the structure of the protein, the embedding from this model is also useful for a bunch of other downstream tasks, such as predicting the property of the protein, such as the thermal stability of the protein, or predicting some additional geometric information of the protein, such as the contact between different residue inside the protein. This model for a single protein prediction will also be a building block for a protein interaction prediction. So one level up is going to be the protein interaction prediction. It takes the embedding from the individual protein representation and perform additional cross-modal kind of transformer to merge the information. And using these tens of thousands of protein complex structure data, we'll be able to train a very nice model for protein complex structure prediction. With this protein complex structure prediction, we'll be able to, for instance, be able to predict what if I have designed an antibody, a protein, and I wanted to bind to a target protein and particular position, what's going to be the position of this binding for this design protein. So we're also using the embedding coming from this embedding this model for predicting the, for instance, whether the protein is going to interact or not, or the affinity of the interaction, things like this. So both the structure of this protein complex or the interaction between the two protein and the interaction strength or the score of between the pair of protein is very important for designing the drug. In some sense, when you design the drug, you want this particular drug to bind to a target protein with a strong enough affinity and in the right position in order to for it to achieve its function, right? So these two models is going to play critical roles in terms of designing drugs once you have already identified the targets in the first process. Okay, based on such a pre-trained language model and connected to structure prediction, so we have been able to achieve quite a improved kind of prediction compared to AlphaFold 2. For instance, here is the results comparing antibody structure prediction. Antibody are a special class of proteins, particularly useful for designing protein drugs, right? And here for the structure prediction, if you measure the error as RMS, the root mean square error, AlphaFold 2 is achieving about 3.5 angstrom, right? In this benchmark data set, we are able to, bring it down further to 1.5 angstrom around. It's resulting in 36% reduction in the error. So TN score is not a measurements. It's about alignment between the predicted structure and ground truth structure. So this pre-trained model is able to also improve significantly in terms of the TN score. The larger, the better. So we also use this pre-trained model for protein complex prediction, right? And for general protein complexes, we are able to bring down the error from around 6 to 7 angstrom to be about 4 angstrom, resulting in 42% reduction in error. In terms of the type of special complex, antibody antigen complex prediction, we're able to obtain even larger, reduction in error, resulting in 89% reduction in error, right? So this is good. We have also used, this is actually a particular example or visualization of the protein complex structure prediction. The protein target is a spike protein from COVID kind of a virus, and then the antibody is this particular antibody in the PDB. And when we use this pre-trained model for predicting the complex structure, it aligns very well with the ground truth. Essentially, it's the purple structure and the blue structure that align very well. The alignment is almost 1, and RMSD is very small. If you use the original AlphaFold tool for the prediction of the structure, the complex structure between these two proteins, then you see that the green one, which are predicted by the AlphaFold tool, actually has quite a big discrepancy with the blue one, which are the ground truth, right? If you look at the predicted antibody structure and look at some specifically important functional regions, this pre-trained model also has a much smaller kind of error comparing to AlphaFold 2. We have also, in some sense, bringing a bit of the parallel computing into the process by parallelizing some of these tensors in this transformer, and we're able to speed up the prediction time by quite a bit as well. Otherwise, if you use the original AlphaFold tool, it might take several minutes to be able to predict the structure and complex structure. So having a more accurate and faster approach to predicting the complex structure, we will be able to screen much more in the computer instead of conducting wet lab experiments. So based on these set of tools, we have actually conducted a pilot study to screen millions of antibodies in silico using the multimer, using the complex structure prediction approach, and then the binding affinity prediction model we have, and obtain some kind of lead molecules, which has quite good in terms of binding affinity value. So the time for the entire AI screening pipeline takes less than a week, comparing to the screening process using animal or fish display library. It's order of magnitude reduction in terms of time, right? So so these are the sort of the 4 nested level of model. The first two is about cellular assistant and cell perturbation prediction. It's about target discovery. The inner 2 level is about protein interaction and protein structure prediction. It's for designing drugs. So in order to automate the process of drug discovery or treating disease, actually you can connect the target discovery process with the drug design process. In some sense, if you have particular disease, the first step you want to do is to find the corresponding target, right? Again, it's a complex process of sifting from a huge combination of potential target down to a few, validated target. So once you have a target, then you want to quickly do some designs of the drug or the protein. Again, it's another problem of sifting from huge designing space to a few potential targets, right? So we want to build a system which can, in some sense, this nested for system, which can link the process of target discovery and drug design process, and in some sense, form a closed loop between the target discovery and drug design as well. There, once you have such system, you'll be able to, perform a new disease and new drug design very quickly, right? So hopefully, I have already exposed to you the current kind of challenge in drug design and the type of data involved in drug design and the scales of the data. And I have also explained a bit about my own exploration of design AI modeling systems, AI models for target discovery and drug design, and the close loop between them. You've also seen a bit of scales of the model. Nowadays, the size of the model are already reaching something like tens of billion or even 100 billion level. And in order to train such a model, in order to do inference in such a model, it's becoming increasingly expensive, and it indeed requires some kind of a holistic design for the system to be able to, really close the loop and let it run very efficiently. So I'd like to end with a question instead of how do we make this system more scalable, right? And with that, I would like to end and welcome you for questions and ideas. Thank you.
Professor Le Song?
Yeah.
We have 2 questions. All right.
Thank you, Professor Song, for the nice talk. I would like to know what's the current level or current capability of the drug design methods. Do we have some successful examples of prototype or drugs already designed?
Yeah, actually, if you look at the AI for drug design company, there are already several companies out there. For instance, Accenture, they have discovered some drugs using AI models, and that drug has reached the clinical trial stage, right? So overall, using AI or complex AI pre-training system for drug design has been very new, right? Most of the company wouldn't be more than 5 years old. Comparing to the entire stage of the 10-year of drug development process, probably AI model is still in the middle. Even the earliest one, the most successful one, is still in the middle. So I imagine in the next 5 years, we will see more and more drug which is coming out of AI-assisted design, right? For me, myself, I've been into the industry for a year or two, right? I'm also at the other stage of designing these AI systems, and we already see these kind of large-scale pre-train model are super helpful in the process of reducing the need for fine-tuned, well-labeled, well-experimented data. Right?
Great, great. Well, maybe let's let the other question go through as well. Yes, go ahead.
Yeah, I want to add a technical question here. So the fascinating talk, it's super exciting here. One technical question. So I noticed that somehow you were able to really now map the multi-omic data, as sequence, protein, and epigenetics, and all sorts of structured data into a transformer-like, deep neural network models or graph nets and so forth. I'm trying to see where is the technical bottleneck. For example, all these data are very heterogeneous, right? What's your representation of this data? Are they each, using a separate representation and, really induce a different type of network architecture, or they have kind of a regularity that, they get a alignable representation or inputs that allow you to scale or to automate?
Right. Thank you for the question. So, so in fact, what I find is this type of question has not been fully explored, right? So at the moment, we are only beginning to actually trying to use these single-cell RNA-seq data. So previous, these data are published on a different paper and curated by different organizations. For the past year, we have been spending lots of effort, trying to put these single RNA-seq data in different places into the same database, right? It involves lots of data cleaning and alignment, right? Both for the alignment between different single RNA-seq data and then the network data. So far, my experience is as we increase the usage of data from less than 100 million to hundreds of million, right? Less than a million to a million. We are seeing very clear improvements. So all of these data are from different sources, different type of cells. The pre-training process is helping a lot. So I imagine that as we fully consume these 100 million data points, and then also explore further the right architecture for this type of data, we can see even more improvements. So one bottleneck at the moment is how are we going to efficiently consume this data? And can we fast enough explore different architectures to extract or squeeze out the information from it? So previously, when computational biologists or bioinformatics experts, they're looking at single-cell RNA data, they have been typically looking at it from dimensionality reduction point, we're trying to remove something called batch effect, things like this. But what we find is when we're doing the pre-training, we don't do too much of that. We still be able to, run some nice representation, which helps downstream tasks such as perturbation prediction. So we haven't got to the stage of, for instance, if we do batch effect removal, will it or not help our model or not? Yeah, there's a lot remaining to be explored, and then the model is big. It's hard to experiment by many rounds at the moment, right? That's why I'm begging for this community to come up with some efficient system or model for this type of data. So one reason standard system cannot feed is also because of the in terms of the sentence is long. It's like 20K tokens in each sentence. And you also have these very meaningful prior network structure in it, which you want to incorporate in this representation learning. Yeah. Hope that gives some, yeah.
Imagery data and other things. I'm just curious, how, right now, in the conventional deep learning world, even combining text and image and co-train a model is already a big deal. And here you definitely have a lot more modality and different forms. I'm just curious, how, we have, a representation, or maybe even just the data representation that allows all this multimodality to enter a single model. It's a very, very fascinating question I want to look into.
Right, that's a very interesting question. At the moment, we have been following the central dogma in biology to ingest this data. For instance, this network, the node has meaning such as protein node, RNA node, and gene node, right? When we are measuring this multi-omics data, right, some are in the gene expression level, some are the protein level, some are the DNA sequence level. So this information could be, mapped to the corresponding node in this network. That way you have a natural way of linking them together. Then on top of it, you can learn representations, right? So here, biology and biological meaning of linking these entities are really important. Yeah.
Amazing. All right. Thank you so much.
Thank you.
All right, thank you. And let's give another round of applause to our speaker, Le. Okay, now I'd like to...